Dear students, greetings to all. In this module, we'll be continuing with our ongoing chapter of biology, chapter 6, molecular basis of inheritance. In this module, we'll be talking about DNA replication, which is defined as a process in which DNA makes copy of itself. So replication is the process of copying of DNA from the parental strand. The parental strand is referred to as the template. Watson and Crick had proposed the semi-conservative model of DNA replication. But then the experimental proof was provided by Matthew Michelson and Franklin Stahl in 1958. They had proved that DNA replication is semi-conservative in manner, where semi means half and conserved means to preserve. So if we see that this is the diagrammatic representation of a double helical structure of DNA, where purple denotes the parental or the template strand and the pink, which is synthesized corresponding to it, is the new strand, which is the complementary strand. So the base pairing is done, like for example, A with base pair with T to form with the help of hydrogen bond. G will base pair with C so as to form a triple bond. And thus a new strand, that is the complementary strand corresponding to the base pairs present on the template strand, is synthesized. The Michelson and Stahl experiment. Michelson and Stahl, they had carried out the experiment by using the bacteria, that is E. coli, and grew them in ammonium chloride having N15, the heavy isotope of nitrogen, and later transferred it to N14 medium, which contained the normal isotopic form of nitrogen. So it was observed that when we talk about it, every generation, the parental DNA, it will get reduced to 50%. So that means if it is 100%, it will move to 50, 25% in the next, 12.5, 6.25, like that. So it was observed that the parental DNA that was having N15, that is the heavy isotope of heavy nitrogen, and that was estimated by using cesium chloride density gradient centrifugation. In the next generation, the replication time required for E. coli, the division time, that is just 20 minutes, and therefore in the next generation, if we consider the first one, there is N15 to be the parental generation, then in the second generation, we'll observe that the DNA which are produced in the daughter cells, they are the hybrid one, having the both N15 and N14, because the E. coli which were grown in N15, they were later on transferred to N14. And later we'll see that the more of the E. coli offsprings, the daughter cells, will have the hybrid and the light strand. That means the N14 gets incorporated in their genetic material. That the estimation can be done with the help of cesium chloride density gradient centrifugation. Talking about DNA replication, it is a process where the DNA makes its copy of itself. Depending upon the type of cell, the DNA replication, like in case of prokaryotes, the DNA is circular, it's small in size, and therefore it goes in for a theta model. Whereas eukaryotic DNA, it's long, it's linear, and therefore it exhibits a Y-shaped fork structure. Discussing about the steps of the DNA replication. So DNA replication, it requires, the first step is the activity of the DNTPs. That means the monophosphate, they'll get converted into triphosphate, like deoxyadenosine triphosphate. The DNTPs, by using the enzyme phosphorylase. So there is addition of phosphate group which takes place in the first step. Next step is to locate the origin, that's the point from where the replication will begin. In case of prokaryotes, there are there is only one origin of replication, but in eukaryote there are many origin of replication. So the initiation of replication, it will take place at one point, which is referred to as the origin of replication. Now we can see that there is the bidirectional replication at one origin, and initiation it will take place at the second origin. So here in the diagrammatic representation is depicted the diagram of a replication fork. So once the origin is determined, there is an incision that develops on the DNA, and then takes place the unwinding of the DNA with the help of the enzyme that is referred to as DNA helicase, or it is also referred to as rep protein, which will bring about the unzipping or the unwinding of the DNA. Now the separated strands, they'll act as the template so that the separated strands do not get coiled to each other again. There is the presence of SSBP, that is the single stranded binding proteins, and these denoted in pink, they are the single stranded binding proteins, which will prevent the coiling of the unwinded DNA onto itself. So they'll make the DNA appear like a replicating fork, Y-shaped replicating fork. Because of this unwinding, there is a tension which is created on the DNA, and the tension, it will be released with the help of the enzyme, which is referred to as topoisomerase, or it is also called as DNA gyrase. So here when the uncoiling takes place, we see that here a tension is created, so that can be relieved with the help of DNA gyrase. Now the polymerization always takes place in the replication takes place with the help of DNA polymerase in the 5' to 3' direction only. It requires the DNTPs as the substrate, and the energy is required in form of ATP. So as can be seen in the diagrammatic representation, the DNA polymerase, it requires a short strand which is referred to as the RNA primer, which is formed with the help of DNA primase, which will carry out the initiation, and then the polymerization is brought about by DNA polymerase. So the one in the synthesis is in the 5' to 3' direction, one strand is continuously synthesized, and that is what is referred to as the leading strand, or the synthesis is continuous synthesis. In case of the other strand, because DNA polymerization only takes place in 5' to 3' direction, the other strand which is being synthesized, that is the lagging strand. It is also called as discontinuous synthesis, and the fragments are called as Okazaki fragments because they are produced as short strands, and then later on they will be joined with the help of the enzyme, which is referred to as the DNA ligase. So the sealing of the fragments will be carried out with the help of the enzyme, which is called as DNA ligase. So here we can see that the primer, it is synthesized with the help of DNA primase, and it gets attached to the 3' end of the template strand, and then synthesis will be facilitated in 5' to 3' direction. Because the synthesis only takes place in 5' to 3', one strand is leading. So this is a leading strand where there is continuous synthesis, and this is a lagging strand where there is discontinuous synthesis. So these short fragments, which are synthesized in 5' to 3' direction, they were discovered by scientist Reizi Okazaki, and therefore the lagging strands are also referred to as the Okazaki fragment in the honor of the name of the particular scientist. And the synthesis is discontinuous mode of synthesis. We can see that the primer, it gets attached, and then the DNA polymerase 3 is carrying out the polymerization. So there are the subunits of the DNA polymerase, which help to the DNA polymerase, that is alpha, beta, gamma, delta, and epsilon, which will help or facilitate the process of polymerization. If the wrong base is introduced, the DNA polymerase will also carry out the proofreading. The lagging strands later on, they will be joined with the help of the enzyme, which is referred to as the DNA ligase. So the DNA ligase will carry out the sealing of the gap between the strands. The machinery and the enzymes which are basically required, like E. coli replicates every 18 minutes. So 2000 base pair per second it comes out to be. In eukaryote, the DNA replication, it takes place in the S phase, and if there is a failure in proper cell division, it will result in polyploidy. So here we can see that in case of the bacteria, so it exploits a theta model where we'll see that replication will proceed. It will start from a particular point and it will proceed in both the direction. So this is the old strand, and that acts as the template, and the other one is the new strand, which is the daughter strand, which is produced on the from the parental strand. In this module, we had carried out a detailed discussion on the replication process, talking about the DNA polymerase. In case of prokaryotes, they are 1, 2, and 3. 1 and 2 are involved in DNA repair, whereas 3 is involved in DNA replication. In case of eukaryotes, when we talk about, so the polymerase, it includes alpha, beta, gamma, delta, and epsilon. So basically the epsilon, that's the it is the controls the HeLa cell, and the delta, it responds for the replication activity. The gamma, that corresponds to mitochondrial DNA repair. Beta is involved in DNA repair, and this alpha, it carries out the initiation of replication. In this module, we had a detailed discussion on DNA replication. Hopefully, you must have garnered all the concepts, the various steps which are involved in the DNA replication. You must have understood them thoroughly. Thank you.